Okay, so this video is going to go over this process of assembling proteins. You've done the reading. You know that this process is actually called infol. The official name that you need to know is translation. That's the full name and the definition of this process of assembling proteins, but really this is just the last part of the process, and we're going to kind of work backwards here. Well, we know that when we're assembling proteins, there are a couple of words that should come to mind, and I'm just going to list them here just to jog your memory. We're talking proteins are made up of amino acids. Amino acids are the monomers that make the polymer that's a protein. Another word for a protein is a polypeptide. Proteins are put together through condensation reactions, which are also called dehydration reactions or synthesis reactions. They produce water when they combine amino acids together. Let's see. And the bond between each amino acid is called a peptide bond. That's where the word polypeptide comes from. When you have lots of peptide bonds, we've got a protein. So that's what we're going to talk about, and we need to talk a little bit about how these proteins are put together and where they're put together. So we're going to start backwards here, and I'm just going to scoot over here. We're going to talk about the proteins. If you remember that in a previous video on enzymes, I believe it was, we talked about a series of amino acids that are linked together. And if these amino acids have a certain sequence to them, these amino acids have a specific sequence, and that sequence is very, very important. We're going to actually do a lesson soon about the sequence of these amino acids and the impact this has on the three-dimensional shape of the protein. Remember that these proteins don't exist on a flat page like we draw them here. They actually exist in real life. So they have three dimensions to them, and that's actually what makes them very unique and interesting. And we'll learn a little bit more about that in class soon. But we need to talk really about this first, is how do the amino acids get put together in the correct sequence. I'm going to put quotation marks around correct because I don't want to imply that there's one right way. It's actually the differences in the sequence that give us variation in the population, but we'll come back to that that idea in the future. So we're going to really focus in on this right here in the sequence of amino acids and how those sequences get put together. So what I hope you know is that this process actually happens in the ribosome. So pick a color here that's pretty mute. Okay, so let's imagine that so I hope you know that this whole process of amino acids getting put together happen in the ribosome. And there are differences in the ribosomes between prokaryotes and eukaryotes. We'll talk about that when we talk about cells in the future. But it's here in the ribosome that the amino acids get put together. And generally speaking, I'm going to use the same color, or similar color combination as in the textbook. Generally speaking, what ends up happening is mRNA, which we'll talk about more. Well, you should have done the reading, but I'll talk more about it in a few minutes. That mRNA comes through the ribosome, and let's say that it's actually traveling to the left. It's traveling in this direction. It's kind of like a conveyor belt, and on it there is a genetic sequence that is being read by the ribosome. So let's say I'm going to put in some tick marks. And for the purposes of this discussion, it doesn't really matter the sequence, but there are specific letters that we use for any kind of RNA molecule. So I'm just going to put some letters here. All right, so I've got AUG, UUA, CGC, AGC, UCU, CGG. That's my sequence. It would continue on, possibly. And as this mRNA sequence comes through, the ribosome is reading it. Now, while that's happening, we've got amino acids that are floating around in the cytoplasm. Now, I might be using some words that you do not remember, so I expect you to look these up. But we've got these amino acids that are floating around in the cytoplasm, and let's think about how they got into the cytoplasm just for a quick second. These amino acids, you got in your body through digestion and absorption. So you ingested proteins. Your body digested those proteins, these polypeptides, into amino acids, essentially doing hydrolysis reactions, breaking up those proteins into individual amino acids so that when they got to the small intestine, they were small enough to be able to diffuse across the cell membrane, across the villi, into the capillary bed that was waiting inside the villi, into the bloodstream. And from the bloodstream, it traveled around the body and most of the time diffuses into each and every single cell in your body. Some cells use them up a little faster than others, but essentially they go into every single cell in your body. They're floating around in the cytoplasm, waiting for the cell to assimilate them. Do you remember that word? Assimilate them, to use them and build them up. And so these amino acids are floating around in the cytoplasm. They're waiting for, I guess I'll use green. Actually, no, I'll use green. They're waiting for what we call a tRNA molecule. And you can draw them whatever way you want, but they're usually drawn kind of like a plus sign. And on the end of them, and on this end of them, where I've drawn these whitish lines, they have some nucleotides, some RNA nucleotides. And I'm going to say that, I'm just arbitrarily, there are going to be lots of different kinds of varieties of tRNAs. I'm going to, let's say, bring in UAC and then AAU. So I'm just randomly putting these, well, not randomly, but I'm just putting these different combinations of letters on. And these tRNA are floating around the cytoplasm, grabbing a specific amino acid depending on its code on one end, bringing in a specific amino acid and waiting for its turn to come into the ribosome and attach to the mRNA. So what ends up happening is that the ribosome reads this sequence in sets of 3 nucleotides. So the first nucleotide here is, this is what we call a triplet code. Notice there's 1, 2, 3. This AUG is the first triplet, then we count the next 3, we count the next 3, we count the next 3, and so on. And we keep reading the mRNA sequence down. Now, of course, there might be other letters up here, but the ribosome is really going to start reading the triplets once it finds this specific one, and this is something important to remember, AUG, that's the start codon. So it starts reading in triplets from the first AUG that it finds. I remember this as AUG. I kind of think this is an abbreviation for augment. And when we augment, we add. And so this is where we start adding amino acids together to make proteins. But what ends up happening, we've got AUG. What ends up happening here is that this tRNA, which has this triplet code is also called, AKA, also known as a codon. And know that codons are on mRNA, whereas on the tRNA, this triplet code is called an anticodon. And there has to be a complementary base pair. So AUG bonds with UAC. UAC will come in and bond with the AUG. Doing so, it's also bringing in this amino acid over here. And then the next codon is being read, UAA. It asks for a tRNA that's floating around in the cytoplasm that has a complementary base pair. So UAA will bond with AAU. Well, look, I've got AAU over here. This AAU is going to come in and bond right here after the UAC tRNA molecule, didn't label that, the tRNA molecule comes in. And for each amino acid that comes in, it adds the amino acid onto the sequence. And so this mRNA will, remember, moves through like a conveyor belt. It's being read one codon at a time, pulling in a tRNA with a corresponding anticodon, which brings in a specific amino acid. When that specific amino acid comes in, we get a certain sequence. So it's really this mRNA that's really key, because the mRNA, put a big star here, the mRNA is carrying a message to the ribosome, which is kind of the machine that's pulling in the tRNA that carry the specific amino acids. And so it's also very important that we have the correct amino acids that are needed by the mRNA for that sequence. I hope that makes sense. Alright, so I'm going to show you an animation that I found online and narrate you through it. But essentially it's this process, but obviously my process is static. The animation is going to move and illustrate these ideas in yet another way. So let me pull that up real quick. Okay, so here it is. It's from McGraw-Hill. And by the way, you can Google McGraw-Hill animations, biology animations, and find all kinds of wonderful resources. I'll let you know that if you click on, if you show the text or listen to the audio, it's going to be a lot more detail and perhaps a little more confusing to you. But it's a good animation showing you what's going on. So let me just point out that we've got down here in red, I hope you can recognize that this represents mRNA or messenger RNA. We've got a sequence of AUG and so on. It keeps going. Notice that there are, we've got the mRNA sequence here. It does have letters, but we're only concerned with genetic information after a start codon. And here's the first start codon, AUG. And this is where the ribosome that's going to come in is going to start reading the sequence and reading this message that the messenger RNA is bringing to it. Now, here is my tRNA. Notice the drawing's a little different than mine, but essentially it's still got this anticodon here. We've got codons here and anticodons based in triplets, and with that specific anticodon comes a specific amino acid. This one is actually, it's labeled F-MET. This is known as methionine, but we'll learn a little bit more about these amino acids in the future. But this tRNA is, T stands for transfer. So this transfer RNA molecule is transferring this amino acid that's floating in this cytoplasm from the food that you digested, waiting to be attached. The tRNA goes and grabs it, kind of like a truck going to get its supply, bringing it to the I don't know, the warehouse, and this mRNA is kind of like the sequence telling it what order to bring the shipments in. Okay, so let's watch this and I'll narrate a little bit. So you got this tRNA floating around. This tRNA finds an AUG just at the same time as the ribosome becomes assembled. Now that might be an interesting idea to you, that you thought the ribosomes were always there. Yeah, they're broken up, and in eukaryotic cells, and this is, I'm just foreshadowing, we actually have two subunits. There's a 50S, and you might have noticed that this is called a 30S. But these two make up what we call an 80S ribosome in eukaryotic cells. But again, I'm just getting ahead of myself. I'm just foreshadowing what these labels are about. We'll talk about that in the cell unit. But if you notice that this tRNA has base paired its anticodon to the codon. Know that A bonds to U and C to G. Now you might know A's, T's, C's, and G's, but this is RNA, not DNA. And we're talking about RNA, we have no T's in here, it's just U's. Okay, so that first tRNA is bonded to the mRNA, and what it's doing is getting the ribosome ready to continue reading the sequence. Now, this is an additional piece of information. There are 3 subunits in a ribosome. We've got a P site. This one over here is called the E site or exit site. P stands for peptidyl, or creates peptide bonds. And then the last one is, this one over here is the activation or active. Yeah, I think this is called the activation site, A site. And this is really the first site. P is the second site, and then the E is the third site. So I think they label them all. Yeah, we got A site all the way on the right and E all the way on the left. And I know this, I just remember like the EPA, like the Environmental Protection Agency. That's my way of remembering it. But if you notice that in the next codon, which is in the A site, a tRNA is being brought in. That complementary base pairs with the anticodon of the tRNA brings in a different amino acid. This methionine is going to create a peptide bond with the proline. Okay, so now we've got a single peptide bond. This is a condensation reaction. Water would have come off. It's not showing you in this model. This methionine is now bonded to the proline. Know that this tRNA now is missing its amino acid. Remember that the mRNA is like a conveyor belt, and that conveyor belt is going to be pulled through the ribosome. So now this lone tRNA is in the E site or the exit site, gets rid of it. We've got another tRNA with an anticodon coming in with a new amino acid, and a new peptide bond is going to be added to this tyrosine. So now we've got a sequence of methionine, proline, tyrosine. The mRNA scoots over, this lone tRNA is going to be kicked out, and a new tRNA is going to come in that has a complementary base pair to GAU, which would be CUA. Here it is. Brings asparagine, and it continues on and on and on all the way down the sequence until the sequence hits what we call a stop codon. There are specific codons, and we'll talk more about this in just a few minutes, specific stop codons that stop the sequence, which is a signal to the ribosome to end the protein production. And at that point, the ribosome falls apart, the protein's amino acid sequence has been made. It hasn't folded up yet, but its sequence has been made, and it disassembles. What's interesting is that these tRNAs, after they've given their amino acids, can be reused. They're going back into the cytoplasm to grab more of that same specific kind of amino acid. So if it brought in proline, it's going out to find another proline. This mRNA sequence can continue and go down to another ribosome. In fact, once a section of it has left one ribosome, it can enter into another ribosome and make hundreds and hundreds of exact copies of the same protein following these same steps. So we really just zoomed in on one ribosome inside of one cell, inside of one organism. But know that there can be hundreds of ribosomes in each cell of an organism, and if we've got trillions of cells, we're talking about this process happening very quickly, very rapidly. Okay? And I think that's the animation. So I wanted to show you this very quickly. This is what we call a genetic code. It looks like a wheel, but I want you to notice that these letters in the yellowish section are A's, G's, C's, and U's. That should be an indicator to you that this is actually representation in which we use RNA, not DNA. If we had T's, we'd see it would be DNA, but because of this U, we know it's which one of the forms of RNA. We have three forms: mRNA or messenger RNA, tRNA or transfer RNA, and lastly rRNA, which is the ribosomal RNA. The ribosome itself is actually made out of an RNA sequence. But that aside, let me tell you that we use the mRNA sequence when we use a codon chart. This is the codon chart. So we use the mRNA sequence. So let's just look at this very quickly. If you remember, our start codon for every single protein is AUG. So let's follow this. We start in the middle with the biggest letters and work our way out. So we start with A, over here, follow the arrow, AUG. Well, AUG leads us to methionine. So AUG that codon on the mRNA always brings in a tRNA with the anticodon of UAC. That anticodon is bringing in methionine. Well, let's look at UCC. Well, UCC brings in serine. So this is essentially how we use a codon chart. You do not have to have this memorized. You just have to be able to use it. And remember, we look at the mRNA codons to figure out which amino acid is coming in. So the part that we're skipping here is the anticodon on the tRNA. But that's pretty easy to do. You should also be able to use an anticodon chart to figure out the amino acid sequence. Okay? So I'm going to put that, let me close that up. And I think that that's the process that all I wanted to talk about for translation. This is the process of putting proteins together. We're going to learn about protein folding next class, but let me just point out that this structure up here on the top right is what we call primary structure, just the order of amino acids. That's all this process is about. The next video is about transcription, which is the process by which this mRNA sequence was made. Okay, so that's what we're going to learn about next. So we're working backwards. We talked about the protein, how the protein's being made. Now we're going to focus on this mRNA and where it came from and how it got its message. Okay, so that's translation. All right.